Hello, welcome to our channel. It's day 1 of the 30-day challenge. The subject is Cell Biology, Membrane Structure. Introduction to the cell membrane. The cell membrane, also referred as the plasma membrane, it is a dynamic and selectively permeable boundary that separates the intracellular environment from the extracellular space. Despite its thickness, approximately to 7 to 10 nanometer, it plays a pivotal role in maintaining cellular integrity and function. Compositionally, it consists of lipid, proteins and carbohydrates. Lipids is roughly around 50% of the total composition. It is forming the structural bilayer. Proteins again almost 50%, and it is facilitating transport, signaling and enzymatic functions. Carbohydrate roughly comes around 2 to 10% and involved in recognition and protection. This composition allows the membrane to be both structurally robust and functionally versatile. Now let's see what is fluid mosaic model. Let's now discuss the fluid mosaic model proposed by Singer and Nicolson in 1972. This model conceptualizes the membrane as a fluid lipid bilayer with protein embedded like mosaic. The key features includes lateral mobility of lipids and proteins contributing to the membrane fluidity, then integral proteins that spans or penetrates the bilayer, then peripheral proteins that associates loosely with the membrane surface, and then carbohydrate attached to the proteins and lipids forming the Glycocalyx. Experimental evidences supporting this model includes freeze fracture electron microscopy revealing intramembrane particles, then FRAP. FRAP is fluorescence recovery after photobleaching, demonstrating lateral diffusion. Third one is X-ray diffraction and ESR. ESR is electron spin resonance spectroscopy. This confirms bilayer arrangement. This model remains the foundational framework for understanding membrane dynamics. Let's understand what a lipid structure is. Membrane lipids are amphipathic molecules, meaning they possess a hydrophilic, means a polar head, and two hydrophobic tails. Hydrophobic means they are non-polar. This dual nature drives the spontaneous formation of bilayer in aqueous environment. The major lipid classes include phospholipids, glycolipids and cholesterol. Each contributes uniquely to membrane structure and function. What I meant by the spontaneous formation of bilayer in aqueous environment is like when this a portion of this lipid bilayer or this composition is put into the any aqueous solution, what happens? The hydrophobic contents of that, whatever the content is, it will come close together, and the hydrophilic will be means directed outwards to the aqueous environment. Thus forms a clear means division between the aqueous environment and the non-aqueous environment. When we understand this, we can understand how the lipid layer is actually forming. Now, phospholipids. Phospholipids are predominant lipid types in membrane. Important variants include phosphatidylcholine, phosphatidylthanolamine. These two includes in the structural role, and then next comes the phosphatidylserine. It is negatively charged, hence localized into the inner leaflet. Then comes the phosphatidylinositol. It involves in the signaling pathway, notably as PIP2. Then comes the sphingomyelin contributes to the lipid raft formation. These molecules confer fluidity and enables compartmentalization within the membrane. Next, let us discuss glycolipids. Glycolipids are confined to the outer leaflet of the membrane and contain sugar residues. We can understand that sugar residue, they are polar, hence it will form bond with the aqueous solution, hence this will be towards the outer leaflet only. We can understand this very clear, and like how the lipid layer will be aligned towards the aqueous fluid, and the remaining portion which is non-aqueous or the non-polar one will be combined together to form into the inner core. So the function of glycolipids are cell recognition, for example AB blood grouping antigen, then protection via Glycocalyx, and cell-cell interaction essential for the tissue organization and immune response. Next comes cholesterol. Cholesterol is present in animal cell membrane and absent in mostly prokaryotes, with the exception of mycoplasma. Its role is to modulate membrane fluidity. At high temperature, it reduces fluidity by stabilizing the bilayer. At low temperature, it prevents gelling, thereby increasing the fluidity. Additionally, cholesterol reduces membrane permeability to small solution. This dual role earns it the title of fluidity buffer, and a concept frequently tested in the CSIR examination. So remember this point. Next comes membrane asymmetry. Biological membrane exhibits asymmetry, meaning lipid composition differs between the two leaflet. Outer leaflet, it's including phosphatidylcholine, then sphingomyelin and glycolipids, and inner leaflet contains phosphatidylserine, phosphatidylthanolamine, and then phosphatidylenol. This asymmetry is maintained by specific enzymes. Number 1, Flippase. Flippase transports lipid from outer to the inner leaflet. The name suggests they flips it from outer to the inner leaflet, and it is an ATP dependent enzyme. Then comes the Floppase. It moves lipids from the inner to the outer leaflet. Then comes Scramblase. It randomizes lipid distribution, and it is calcium ion activated. Importantly, phosphatidylserine exposure on the outer leaflet is a hallmark of apoptosis, detectable using annexin 5. Remember this point. Then next one is membrane proteins. Membrane proteins accounts for approximately 50% of the membrane mass and are classified as integral or intrinsic protein, then peripheral or extrinsic protein, and lipid anchored proteins. Integral proteins, like they span the membrane as alpha helix or beta barrels. It serves as channel, receptors, pumps and enzymes. It requires detergent for removal from the lipid bilayer. Then comes peripheral protein. Peripheral protein means periphery, extrinsic means outwards. It is loosely attached via ionic interaction or cytoskeletal links, and it is removable by salt or pH changes from the lipid bilayer. Then comes the membrane carbohydrates. Carbohydrates are exclusively found on the intracellular side of the membrane, forming glycoproteins or glycolipids. The function includes protection via Glycocalyx, recognition by lectins, adhesion and immune response facilitation. Next comes the membrane fluidity. Membrane fluidity influenced by several factors. Factor number 1, fatty acid saturation. Unsaturated means it is having increased fluidity, and saturated means decreased fluidity. We can understand saturated means the bonds are all means all bonds are 3 bonds of the carbon is full within each other, so the fluidity is decreased, and unsaturated is more flexible, hence high fluidity. The next one is chain length. Shorter chain length, higher the fluidity. It is just a common sense. When the chain is shorter, fluidity is higher. Then temperature, higher temperature, fluidity increases. Then cholesterol composition acts as fluid regulators. Cholesterol acts as a fluidity regulator. Then protein contents. High protein density means the fluidity will be less in those portion where the protein is present, and hence high protein density leads to decreased fluidity locally. Then comes the membrane domains. Membrane contains specialized regions like for example lipid raft, caveolae, then tight junction and desmosomes. Lipid raft enriched in cholesterol and sphingolipids. It is less fluid and it is involved in signaling and endocytosis. Caveolae, it's a flask-shaped invaginations containing caveolin, and it is involved in mechanosensing and cholesterol transport. Tight junctions and desmosomes, it facilitates cell adhesion and barrier formation. Now let's understand membrane permeability. Permeability hierarchy. Hydrophobic molecules, it is like oxygen, carbon dioxide, it is highly permeable because it is hydrophobic. It can easily pass through the lipid bilayer. Then comes small uncharged molecules that like H2O, urea, they are moderately permeable. Large uncharged polar molecules like glucose, it is poorly permeable. Then ions like sodium, potassium, calcium, it is very low permeability. Hence the permeability hierarchy comes like from high permeable to low permeable, hydrophobic molecules, small uncharged polar molecules and large uncharged polar molecules. Then comes the ions. Ions are charged particles and hence it has very low permeability. Water crosses membrane via aquaporins and partial diffusion. Now let's understand techniques to study membranes. Key investigative techniques are FRAP. FRAP measures the lateral diffusion of membrane components. Then comes freeze fracture EM. It reveals protoplasmic phase, exoplasmic phase and integral membrane proteins. Then comes the patch clamp. Patch clamp quantifies ion channel activity. Then fluorescent labeling, it tracks compartmentalization and protein dynamics. Now let's understand the high yielding concepts according to the CSIR UGC NET examination. So let's summarize it. Like lateral movement is rapid, flip-flop is a slow and enzyme dependent process. Then PS exposure signals apoptosis. It is a signal of apoptosis. Then comes the membrane thickness. It correlates with fatty acid chain length. Then GPI anchors localizes protein to the outer leaflet. Then hormone receptors are often found in lipid rafts. Then cholesterol is absent in bacteria except mycoplasma. Then comes barrel proteins are found in mitochondria, chloroplast and bacteria. Then membrane diffusion and membrane fusion and fission requires snares and dynamic. These are the frequently tested part in part C of the CSIR examination. Ensure the conceptual clarity and application. And that concludes our session on membrane structure. I encourage you to revise these concepts using diagrams and practice MCQs to reinforce your understanding. In our next class, we will be exploring membrane transport mechanisms, and it will include passive and active transport, facilitated diffusion, and endocytosis. Thank you, and stay connected with your preparation and be consistent. Thank you.